Hello, my name is Kevin Ryan. I'm a PhD student at the University of Galway, and this presentation is for my poster presentation at Transmed Community of Special Interest at ISMB ECCB 2023. The title of my poster is Transcriptomic Characterization and Identification of Potential Neoantigens in Cancer-Associated Fibroblasts in Breast Cancer, and I'm supervised by Dr. Laura Barclay and Dr. Philip O'Brien. As a background, cancer-associated fibroblasts are an important part of the tumor microenvironment, and they are of great interest in cancer therapy. There's two big challenges when it comes to targeting CAFs therapeutically, one being a lack of CAF-specific markers, meaning that off-target effects often result from CAF depleting therapy. Another challenge is CAF heterogeneity, meaning that CAFs can have tumor restraining as well as tumor promoting roles. The purpose of my project is to try to address some of these issues by identifying CAF-specific neoantigens using whole exome and RNA-seq data, hopefully addressing the issue of the lack of CAF-specific markers, and then all the while taking the heterogeneity of CAFs into account by characterizing patient-specific CAF heterogeneity using RNA-seq. These are the methods that we used. Samples were taken from tumor and the adjacent normal tissue of patients undergoing surgery for breast cancer. And then this tissue was excised and digested, and then plated. A suspension was plated to encourage the growth of fibroblasts. So for each patient, we had a tumor and a normal sample. The tumor sample is our CAF sample. The normal sample is our tumor adjacent normal fibroblast sample. Then RNA sequencing was carried out in 12 patients. Whole exome sequencing was carried out in 6. The RNA-seq, we could carry out a differential expression analysis using DSeq2, the CAF subpopulation deconvolution using CyberSort, and try to identify fusion neoantigens using NeoFuse. Using the whole exome sequencing data, we could identify missense mutations, and then using the landscape of effective neoantigen software or lens, we could identify putative neoantigens resulting from single nucleotide variants. Note with these patients is that 11 of the 12 had luminal A breast cancer, whereas one patient had triple negative breast cancer. When differential expression analysis was carried out, we found a number of differentially expressed genes. And then when we looked at the pathways, the upregulated processes in our CAFs tend to relate to things like the actin filament-based processes, suggesting a myofibroblastic phenotype, and then the downregulated processes tended to involve the immune system. It suggests that in this case, our CAFs are playing a myofibroblastic and immunosuppressive role. But we wanted to get a better look at the heterogeneity of our CAFs in these samples. And to do this, we carried out a cell type deconvolution. So we took data from 3 of 4 previously described CAF subpopulations that are publicly available. We took this data and combined it with our own data. So we processed all the data using NF-core RNA-seq, carried out batch correction and deconvolution of subpopulation proportions using CyberSort. When we did this, we found in the different patients, we had different proportions of the different CAF subpopulations. And then when we compared the proportions in our CAF versus our adjacent normal samples, we didn't find a significant difference in the different CAF subpopulations. We then used NeoFuse to predict fusion neoantigens. And when we did this, 4 of the 12 patients were predicted to have one CAF-specific gene fusion, resulting in at least one fusion neoantigen. We then went to look at neoantigens resulting from missense mutations using the landscape of effective neoantigen software or lens. When we did this, we found a number of potential neoantigens. So all of them were actually seemed to be expressed. So the TPM of greater than 33, and all of them seemed to be similar to known immunogenic peptides. However, none of them had a favorable binding stability, predicted binding stability, and only one of them had agrotopicity of less than 0.1, which is the ratio of the predicted binding affinity between the mutant and the wild type peptide. And then we looked at the mean predicted binding affinity rank of these three tools, MHC Pan, MHC Flurry, and NetCTL Pan. Only one of the tools had a favorable actual binding affinity rank. So to summarize, we identified differentially expressed genes between CAFs and adjacent normal fibroblasts. We used CyberSort for the deconvolution of CAF subpopulation proportions in our patient CAF samples. We found a small number of potential neoantigens resulting from single nucleotide variants and gene fusions. Future work will involve the use of lens to infer neoantigens resulting from insertions and deletions, aberrant splicing, and endogenous retroviruses, and more. And we hope to repeat this work in the context of lung cancer with the addition of single-cell RNA-seq data. And here are my references. And I'd like to thank my funding agency for Science Foundation Ireland for funding this research and making it possible. Thanks very much.